So we're going to look at some cases where we've been able to identify changes in the Hox genes and how that has impacted the diversity of animals, and particularly with some specific groups. A few lectures ago we actually went over some of these, so a little bit of review, but a lot of new stuff here also. So let's jump in. So here's a diagram showing all of the diversity of Hox genes. So you've got your phylogeny. You memorized the major groups here, so hopefully you recognize some of these, right? You've got your deuterostomes here, your protostomes down here, with a few more groups than I asked you to remember, but remember there are about 30 different animal phyla, and so we only have even here a small number, but the big ones with the most species. And then as our outgroup here, the Cnidaria, we could even put one more down here, the Porifera, right, the sponges. Notice the Cnidaria have a fairly simple Hox cluster. So that is the first big evolutionary event in the history of animals with regards to the Hox gene, as we go from a rather simple, maybe 2 copies of the Hox genes, to a much more complex one. And so we're going to look at that here in a little bit. But before we do, let's look at a case study with Drosophila. Drosophila is an important model organism. We know a lot about its genome. We know a lot about how it developed. So it's a really great organism to use for this sort of thing. And so I'm going to focus in particularly on this gene called HOX3. So it's the one in the orange color here. Now notice it's a standard Hox gene in the vertebrates amongst most of the other protostomes. It's a standard Hox gene also, but among the arthropods it has a little bit of a different function. So in Drosophila, HOX3 is actually duplicated. So most organisms only have a single copy of this one, but the fruit fly actually has 2 copies. And both of them have a very, very important function, but it's not the typical standard Hox function. So I want you to think back. Remember how we defined that cascade of genes during early development? You start with the maternal effect genes, the gap genes, the pair rule genes, and so on down the line. You get to the 5th level, and you have the Hox genes. And the ancestral, the canonical, right, or the traditional role of those Hox genes is to pattern different regions of the body to help determine what's going to be the thorax versus the head versus the abdomen. Okay, and so that's what Hox genes are doing most of the time, but there are some exceptions to the rule. And HOX3 in arthropods, and specifically in Drosophila, is one of those where we actually have extra copies. So HOX3 sometimes goes by other names because these were identified before it was recognized as a homologue of HOX3 in other animals. And in the fruit fly we have one that we call Zen and one that we call bicoid. And you guys already know bicoid. So remember, bicoid has taken on this brand new function in the fruit flies. In most arthropods, it's a very different gene and different process that determines where the head is going to be. But bicoid is one of those very, very early maternal effect genes that tell a developing fruit fly embryo where its head is going to be. And this is a fairly recent innovation in the history of the fruit flies. It's something that their ancestors didn't do, and so it's really a unique feature for the fruit flies. So it just kind of shows you how you can have changes to these developmental genes. Here we've got a little bit of duplication coupled with some diversification and expansion of function to do something that's brand new. Okay, so HOX3, which one of the descendants, the paralogous descendant of Zen and bicoid, now has that new role. So just remember that, and know that as an example of some of the diversity that can happen in Hox genes. So though there is a fairly standard function for Hox genes, and that's fairly widely distributed across the animals, there are some pretty cool and interesting exceptions to that. Okay, the other one we're going to look at as Fitz. Fushitarazu, Fitz is just a nice abbreviation. Fushitarazu is another Hox gene, and so if we come back here to look at its homologues here, it is HOX6. And so again, canonical Hox function for these across most organisms, but we've got this duplicated version where Antennapedia is one of the descendants from that Hox gene 6 in the ancestor, but we've got this extra one, so Antennapedia is the one that maintains the canonical Hox function, patterns the head as the name implies. But This other one, Fushitarazu, now has a different function, and it's actually, again, upstream in the fruit flies. It has some slightly divergent function across other insects, and so people are still trying to figure out all of its diversity of range, but it's a little bit more flexible than most of the Hox genes. So once you have this basic set of Hox genes, and remember we call this our Hox gene cluster, so that basic cluster will often maintain integrity, but when we get extra copies we can do something brand new. And there is some possibility for moving it or moving things around, losing one gene or another, and still maintaining integrity of development. But by and large, it's very, very conserved across wide spreads, with some few interesting examples like these ones we talked about for fruit flies. So let's look at 3 major changes. This is a little bit of review. We hit these in a previous lecture, but we're going to reinforce them. They're important. Make sure you know them. So let's start with the first major event. So we're going to come back to Cnidaria. These early, simple, basically only one major tissue type, no complex tissue layers, not any complex organization in the Cnidaria. These are the jellyfish, right? And so their Hox genes are correspondingly simple. They just don't need a lot to make their bodies because they don't have complicated bodies. And what we see, as demonstrated here with the Lophophorate, a group we didn't learn about, but it's one of the protostomes, we get a much, much more complex Hox cluster in the Bilateria. So this is just representative. You'll see some of the other Bilateria here also. So that very first thing, they're at the base of the Bilateria. So with deutostromes, protostomes representing here. This is kind of the simpler, but maybe more what the ancestor would have looked like. The Lophophorate has kind of maintained that, but still much more complex than the Cnidaria. So that happens at the base of all of the Bilaterians. Okay, so somewhere in their ancestor they get lots of duplication events, and we get a complex Hox cluster. And so this is, and again, notice that's fairly similar. There are a few differences there with the Lophophorates, but fairly similar to what's going on in the ancestral Hox cluster, where we have these 10 Hox genes. And again, in insects, this one is diverged and this one is diverged, and so we do get differences, but that's the ancestral function. And these 10 Hox genes then go on to be the basic blueprint building blocks from the different body regions amongst all the organisms. So number 1, we get kind of this full complement that allows for the complexity of Bilateria. The next thing here, we're going to look at number 2. So again, reminder down there, number 2 is only in the deuterostomes. And so in the deuterostomes, we get this expansion of the posterior Hox genes. So notice in the protostomes, represented here by the Lophophorate, we only have 2 of these posterior Hox genes. So remember, the Hox genes are collinear. This one here in red is in charge of patterning the head, and then the hind part of the head, and then the thorax, and then so on back until we get to the abdomen. And so the protostomes have a fairly simple complex of genes that patterns their abdomen. So notice they're basically 2. Occasionally there's a duplication. There's some that have been lost. And the nematode has 1 extra one. That's a little bit of an exception to the rule, but it's rather simple. But then notice the deuterostomes. The echinoderms have either 1 or 2. There's variety and diversity there. So at the minimum there are 3, but in some groups there are even 6 or 7 as represented here. And the higher vertebrates, we've got 6 to 5 different sets of Hox genes. And then of course this entire duplication, which is going to be number 3. And I forgot to put a number 3 there, but we'll just look at it on this new slide. So the third major innovation in the Hox genes is the duplication of the entire cluster in the ancestor of all of the vertebrates. And there is a lot of variation here. So most tetrapod vertebrates have 4 of them, again with some losses of individual Hox copies in these different clusters. But notice we've got dozens and dozens of Hox genes, where the cephalochordates, the echinoderms, and all the other vertebrates only have this one major cluster. So this allows for lots and lots of different developmental diverse processes to occur. Some fish even have more than 4, so that's why it's kind of, instead of just saying a 4-fold duplication, which generally holds, that's probably ancestral form, but fish have even more than that. Okay, so we go from this simple ancestral one represented here by the fly, right? This is the ancestral for the Bilaterians, not for all animals, but here it is represented by the fly, to this much, much more complex form. And then, of course, in the fly, we get these interesting stories. And there are dozens and dozens of little interesting stories. So we could come back here and tell a story about the Onychophora and how they've kind of duplicated a little bit and lost some, how we get extra copies here and there. And even if we zoomed in on some of these, looked at smaller groups within the nematodes or within the mollusks, for instance, we would see other small stories, but these are the big ones. So remember 1, 2, and 3. Remember what those are and what groups they apply to. So number 1, the overall copying and diversification in the ancestor of the bilaterian. Number 2, the expansion of those posterior genes. So we get many duplications, meaning more posterior genes, and that's just in the ancestor of the deuterostomes. And then finally, in the ancestor of the vertebrates, we get this fourfold duplication and then eventually even more duplications in some groups. So building, building, building all of this developmental gene complexity, which allows for the complexity of these different organisms. Okay? And so we can look at individual cases like the UBX case that we talked about, and we can even map those to very specific residues. So we can say the changes here in this one gene, in this one part of this gene, are what leads to this major innovation in organisms. And then we can begin to do this in very, very fine detail. Okay? So let's see if we can extrapolate whether or not these diversifications at the genetic level lead to diversification at the morphological level. Okay, so question number 1, and then we'll look at 2 cases that test this statement. Does expansion of the toolkit result in morphological complexity? And so we've already looked at 3, but 2 of them that stand out especially. So let's look at those 2. Case number 1, the cnidarians, right? The very early animals were like cnidarians. And then we get the evolution of the bilaterians. And of course, at that base here, you get that expansion of the Hox tool set. And all of these bilaterians are incredibly complex compared to the cnidaria and the porifera. So this holds true. This is one bit of evidence that, by any measure, right, these simple cnidarians with simple Hox genes, and then the more complex bilaterians with complex Hox genes, right? So here's some example of cnidaria. We have anemones, we have hydra, the jellyfish. There's some other groups also. And they've been important historically and are still somewhat important in many ecosystems, but are much less complex than the bilaterians. Okay? And so if we look at bilaterians, scorpion is just one example. We could put dozens and dozens of them up here. But we see all of these things. The bilaterians have more tissue layers. They have a coelom, which helps to protect and buffer their inner organs. They've got a gut that is all the way from the beginning to the end, where we've just got kind of a simple sac or like dead end digestive tract on the cnidarians. The body axis, right, which is bilaterally symmetrical, and that adds complexity. And then finally, maybe most striking is their neurological complexity. So by any measure, dozens and dozens of different criteria, these guys are more complex. And so almost certainly the Hox gene diversification is responsible, maybe even directly responsible, or at least largely responsible for the complexity of the bilaterians. So case number 1, yes, right, we've got support that does seem to say that when we get diversity of the Hox genes, that leads to more morphological complexity in those organisms. Okay. Case number 2, let's look at the vertebrates. And remember, vertebrates have this fourfold and eventually more in some of the fish, but this fourfold duplication of the entire Hox complex. Right? So there it is. I've just highlighted it there on our little phylogeny. And you can see the diagram here showing you that, right? So there you go, more complexity. So does that lead to more morphological complexity? And the main component here, although we can look at a few others, but the main component is the central nervous system. Brains of vertebrates are by far way, way more advanced and more complex than any other organism, even the decapod mollusks. So those are the, octopod mollusks, right? The cephalopods, the octopus, the squid. Those cephalopods are very, very smart, but their brains, it's one of the amazing things about these guys is that they can be so smart despite having a relatively simple, morphologically simple brain. Okay, So a very, very complex central nervous system amongst the higher vertebrates. Also, there's a lot of ecological things that they're very, very complex in the way they interact with their environment. So those two primary measures, but probably some others that you could throw in there if you thought hard about it. And sure enough, these vertebrates, both historically, right, with extinct lineages, and today, and we could put up more than this, but I've got fish, you know, you could look at mammals, you could look at birds, right, the last living remnants of the dinosaurs, and those are way, way more ecologically complex, and also morphologically, especially in the brain, but very more complex. So again, both of these case studies where we get a duplication of the Hox genes, diversification of them, that tends to lead to more morphological complexity. These Hox genes are the basic building blocks of organisms and how they pattern different body regions. So when you have more of them, you have more potential for doing different things. And so that's basically, in a nutshell, why animals are able to be so successful and so morphologically complex. Okay, one other question. Is biodiversity, so species count, is that the result of this Hox gene diversity? So if I get more and more Hox genes, does that mean I'm going to have more species? And the short answer to this one is no. And we could look at several examples, but here's one we've already talked about, right? Arthropods, by far the most diverse species, biodiverse, and speciose group of organisms on Earth. There are more named ones and they're small, so there are many, many more out there that we haven't named, right? There aren't that many monkey species left to discover, but there are tens of thousands, maybe even into the hundreds or millions of beetle species that are undescribed. Okay, so incredibly biodiverse. Their closest relative are the onychophorans, which have just a few dozen species. So these guys have about the same level of diversity. If you look at their Hox complexes, they're about the same, right? So diversity is really more of finding an ecological niche, finding maybe a new innovation. So they can use the same Hox genes, but maybe reworked and networked in different ways to build a little bit of more of a complex body. So biodiversity, we're just going to say no, right? 80% or so of all described species are arthropods, or only a handful of onychophora. Here's just some of that arthropod diversity. Aren't these guys gorgeous? So this you might recognize from the Harry Potter movie. This is a vinegaroon or whip scorpion type of arachnid. This is a crustacean praying mantis shrimp. We've got a giraffe beetle here and centipede, right? And so all of this diversity and species and success is built off the same basic Hox complexity as we have here. So biodiversity is not really a one-to-one correspondent with Hox genes. Or maybe a better way to put it is having a biodiverse group does not necessarily mean you're going to have more complex Hox genes than other groups. Okay, so that's it. Remember, I'm breaking these lectures, so this is 2.6, but this is just part A. You've got an SLO that goes along with it. I've got the shortened version of the PowerPoint available in the PowerPoint folder, and also the bigger long one, which has both parts of 2.6. Also, please read paper number 7. I will post a few guide questions to paper number 7 in the next day or so, on maybe the morning of the 24th of March. So take a look at that. You don't need to read it in a ton of detail, but skim over it. I'll post some guide questions so you can kind of get an idea of what the key ideas are coming out of paper number 7.